The overall goal of this procedure is to observe the ultrastructure of megakaryocytes located within the mouse bone marrow and to quantify their different maturation stages using high-resolution transmission electron microscopy. The main advantage is the direct examination of megakaryocytes in their native environment, which differs from in vitro cultured megakaryocytes that, as we know, do not reach the complete level of maturation.
After harvesting the tibia and femurs from 12 to 18-week-old C57 black 6 mice according to standard protocols, use a sharp razor blade to remove the epiphyses from each bone. Holding each bone with tweezers, insert a 21-gauge needle attached to a 5 ml syringe filled with cacodylate buffer into one end of the bone, and flush the bone marrow into a 15 ml collection tube containing 2 ml of fresh cacodylate buffer. Immediately after flushing, use a plastic pipette to transfer the bone marrow cylinders into 1 ml of fresh glutaraldehyde fixative solution for a 60 minutes incubation at room temperature. For embedding of the bone marrow in agarose, wash the fixed samples in fresh cacodylate buffer and use a plastic pipette to carefully transfer the bone marrow to a glass slide. Using a warm pipette, quickly apply a drop of 2% liquid agar to the bone marrow cylinders and immediately place the slide on ice for 1 to 2 minutes. When the agar has solidified, use a stereo microscope and a sharp razor blade to discard the extract.